Inside a single-celled bacterium runs a specific, highly ordered chemical operating system, dictating exactly how it survives and reproduces. A towering, centuries-old redwood tree operates on that exact same chemical software. Despite the immense difference in scale, the underlying programming language used by both organisms is identical. This shared biological hard drive is deoxyribonucleic acid, or DNA. DNA is the molecule found in nearly every cell that contains the biological instructions making each species unique. The diversity of life on Earth results from different sequences written in this single ancient code. Zooming into the nucleus reveals how this information is stored. It takes the shape of a twisted spiral staircase, a structure known as a double helix. The outside edges of this molecule form the handrails of the staircase. They are built from alternating sugar and phosphate chemical groups, creating a sturdy backbone that holds the entire structure intact. Spanning the gap between these handrails are the rungs of the ladder. These are nitrogen bases, and they come in 4 types: adenine, thymine, cytosine, and guanine. These 4 chemicals act as the literal A, T, C, and G alphabet of the biological code. These bases connect across the center of the helix using strict chemical logic. Because of their specific shapes, adenine only pairs with thymine, and cytosine only pairs with guanine. A human genome consists of about 3 billion of these individual base pairs. The precise sequential order of these chemical letters provides all the specific instructions needed to build and maintain an organism. Within those 3 billion letters, specific sequences are grouped into functional sections called genes. Each gene contains the instructions to produce one specific biological trait or protein. To protect these instructions, DNA remains inside the cell's nucleus. This central vault keeps the master blueprint secure, preventing it from leaving the nucleus. This creates a logistical problem. The cell must find a way to get those instructions out to the manufacturing areas where biological construction takes place. The cell solves this through transcription. Enzymes open the DNA helix to read a section and create a temporary copy called messenger RNA, or mRNA. This intermediary molecule conveys the genetic data out of the vault. mRNA carries the required biological code to the rest of the cell, while the original DNA sequence stays protected inside the nucleus. Once outside the nucleus, the messenger RNA travels to the cell's manufacturing hubs, the ribosomes. Here, translation occurs. The ribosome reads the RNA sequence, translating it into a chain of amino acids. The ribosome links these building blocks in the exact order dictated by the RNA, folding into a specific 3-dimensional protein. These proteins are the physical laborers of the organism. They build the hardware, from muscle fibers to enzymes, that keeps the creature alive. As an organism grows or heals, it must create new cells. Each new cell requires an exact copy of the original biological blueprint. To copy itself, the DNA molecule unzips down the middle, pulling the paired bases apart and leaving 2 single strands exposed. Because A only pairs with T and C with G, free-floating chemicals slot into designated places on the template, rebuilding 2 identical double-stranded molecules. This structural templating system ensures that billions of letters of biological information are passed precisely from one generation to the next. For billions of years, this operating system ran in total darkness, unknown to the creatures it built. That changed in 1953. By studying X-ray diffraction patterns and building models, researchers deduced the double helix structure, proving it could carry biological information. Decades later, an international scientific effort successfully mapped the entire human genome, recording all 3 billion letters of the Human Instruction Manual. Humans share more than 99.9% of their DNA sequence. By analyzing the tiny fraction of letters that vary between individuals, we can trace human ancestry and evolution. Scientific focus has shifted toward active intervention. Researchers are now developing tools to write and edit the DNA sequence directly. By altering specific sequences, doctors can now correct biological errors at the source before they manifest as inherited diseases. Even as our ability to manipulate the molecule grows, the underlying mechanism remains the same. The 4-letter language dictating modern breakthroughs is the exact one used by ancient bacteria. It is an unbroken chain of chemical data, forever connecting humanity to every other living thing on Earth.